Hello everyone. Today we are going to learn about endospore staining versus capsule staining. Okay, so in this video we'll focus on the differences between the endospore staining and capsule staining. See here in our first difference brings the purpose. Okay, so our first aspect is purpose. So first in the endospore staining, as we know that most vegetative cells, they are stainable, right? With standard aqueous dye solutions, they are stainable. But these endospores, they possess a permeability barrier that prevents dyes from entering. Okay, so because of this permeability barrier of the endospore staining was developed. Right, so this endospore staining is used to identify the spore forming bacteria. For example, Bacillus cereus and Clostridium. Okay, Clostridium botulinum, for example. So this endospore staining is used to identify the spore-forming bacteria. The bacteria, those who have spore, and these spores, since they have the permeability barrier problem or complexity, that is why this endospore staining was developed. Now, this endospore staining is also used to detect the presence and location of the endospores in the bacterial cells. Okay, now the location of the endospores in the bacterial cells can be different. So here in this bacterial cells, you can see that the endospore is at one end of the bacterial cell. Okay, so that is why this is endospore. Okay, since it is inside the bacterial cell. If the spore is outside the bacterial cell, then it will be called as exospore. Okay, so all this type of detection can be done using endospore staining. Now in the capsule staining, this capsule staining is used to detect the presence of a capsule. What is this capsule? It is a polysaccharide or a protein layer that surrounds some bacterial cells. Now you need to remember that all the bacterial cells does not possess capsule. Okay, this capsule is possessed by some of the bacterial cells only, and this capsule is made up of polysaccharide or a protein layer. Now this capsule staining is used to identify the capsule producing bacteria like, for example, Klebsiella pneumoniae and Streptococcus pneumoniae. Now here in this diagram you can see the type of the capsule producing bacteria and how they are looking under the microscope. Now here is a note that we need to remember is that these capsules, the bacteria, those who have capsules, the function of this capsule is that it will allow the bacteria to evade host immune system and enhance virulence. Okay, so majority of the bacteria having capsules are pathogenic in nature. Okay, they evade the host immune system easily and they enhance virulence after getting inside the host immune system. Okay, so identification of this capsular bacteria becomes very important, and for this only the capsule staining was developed. Okay, now here there is a point. There's this endospore staining and capsule staining, both are a form of differential staining. Okay, as we have learned in our previous video. So both are a type of differential staining. That means differential staining allows the structural visualization of the bacterial cells. Okay, so here we can do in both the staining methods, we can see the structure of the bacteria, and also we can understand the structural composition of the bacteria. Okay, so this is the these two are the both differential staining method. In our previous video, in our simple staining versus gram staining, both the methods were different. One was simple and one was differential. Whereas here in this video, we will talk about the two differential staining method completely used for different purpose. One is endospore staining, another is capsule staining. Okay. So our second aspect is this reagents. What are the reagents that are used in this method? Now first for the endospore staining we need a culture of the a bacteria we have who have endospore, obviously, for example Bacillus cereus or Clostridium botulinum. Next we need the stain malachite green and safranin. Then we need Bunsen burner, staining tray, inoculating needle, water bath and microscope. So these are the requirements. For capsule staining, we have culture of the capsule containing bacteria, for example, Streptococcus pneumoniae. Then we need stains that are crystal violet and nigrosin. Then we need slides, inoculating loop, and microscope. Okay. So now coming to the procedure. Now this will show the pictorial flowchart of the endospore staining. Okay. So here is the flowchart of endospore staining. So first you need to prepare the smear as we did earlier, and then you have to heat fix it. Okay, so you heat fix the smear, and then after that you put the 0.5% malachite green over the smear. Okay, you just flood the slide with 0.5% malachite green after heat fixing. And after that, what you do, you put this slide over the water bath for 5 minutes. Now, what will water bath do? Water bath will allow the smear to steam, right? And the steam will allow the stain to get inside of the bacterial cells. Okay, since endospores are resistant structures, so they will not retain the stain easily. That is why the water bath is used to enhance the penetration of the dye inside the endospores. Okay, so this steaming method is very important, very critical part of this endospore staining. Okay, now after that you wash the slide with distilled water. After 5 minutes, you wash the slide with distilled water, and after that, you add the counter stain saffron in for 30 seconds. You keep it for 30 seconds, and after 30 seconds, you again wash it with distilled water. After that, you air dry the slide, and after that, you can observe it under the microscope at 40x or 100x using oil immersion. Okay, so this is the complete pictorial flowchart of the endospore staining. Now coming to the capsule staining. Okay, so here in this capsule staining, the methods are quite different. So here in the capsule staining, first what you do, you take a grease-free slide. How to do the grease-free? After cleaning your slide, you take an alcohol in your cotton and you grease free your slide. You just wipe your slide until there remains any distortion on the slide. Okay, so that when you make the smear, your smear will appear in a continuous manner, not in a discontinuous manner. Okay, so you take the grease free slide here, and then what you do, add nigrosin ink at one edge of the slide. Okay, as shown in this picture. Then after that, what you do, you take the bacterial culture using this inoculating loop, and you mix this bacterial culture in this nigrosin ink, in this dye, and you mix it properly. Okay, you put the inoculating loop which have the culture in this drop, and you mix it properly gently. Do not make a round circle, just mix it inside the drop. And after that, what you do, you take another clean slide and you put this clean slide over the top, and you drag the second slide over the first one, making a thin film. Okay, just as we do in our negative staining method. So this capsule staining uses the negative staining only. So here what you do, you take another slide, you put it over the drop of the nigrosin ink, and you drag the second slide over the first one, making a thin film, continuous thin film. Okay. After that, you allow the film to dry, and then you can observe it under the microscope at 40x or oil immersion. Okay, so this is the pictorial flowchart of the capsule staining. Now the details of the procedure of capsule staining as well as for this endospore staining has already been covered in our capsule staining and endospore staining video. If you haven't watched it and you want to have the detailed procedure for your understanding, you can check the description below. Now, what are the results that we can expect from this experiment, right? So here in the endospore staining, these endospores will appear bright green in color, while the vegetative cells will stain pinkish, will give pinkish color. Now in this picture, as you can see, here the vegetative cells are pink in color, whereas some of the bacterial cells that contain the endospores, these endospores are bright green. Here all the bacteria does not have endospore, as I said earlier. Okay, only some of the bacteria will have the endospore. So from this staining, or with the help of this staining, you can also identify the spore forming bacteria from the non-spore forming bacteria. Okay, now let us see another picture. So this was an image of Bacillus subtilis, and this is of the Clostridium botulinum. Here the Clostridium botulinum bacteria, you can see it has a different shape from that of the Bacillus subtilis. However, you can see the endospore, right? Here you can see the endospore, which are stained green in color, whereas the vegetative cell are stained pink in color. Okay, so this is the observation that we can see under the microscope. Now coming to the capsule staining, this capsule bacteria, what they do, they will not retain any stain and they will appear translucent against a dark background, just like this. Okay, here you can see this is the dark background and these capsules have not taken any stain and they are appearing as a translucent or halo. You can see a halo or clear region surrounding the bacteria. So this shows the presence of the capsule. Here also you can see that some of the bacteria have the capsules and some of the bacteria don't have the capsule production. Okay, so from this staining method also you can identify the bacteria that are having capsule, the capsular bacteria, from the non-capsule producing bacteria. Okay, so these are the types of the differential staining method that we can see. Here the structural visualization can be clearly done. Here you can see the shape and structure of the bacteria as well. Okay, so these are the results that we can expect. Now coming to the mechanism behind the this type of staining. Okay, so first in the endospore, as we have learned that the barrier, the permeability barrier which we have seen in this endospore. Okay, this barrier develops as the spore coats. Why they develop? Basically they develop because in order to protect the cell's DNA and vital proteins from adverse environmental conditions. Okay, now some of the bacteria, what they do, they form spore coats, they develop spore coats. Now these spore coats are one type of the barrier which are very impermeable. Right, why they develop the spore coats? In order to protect the cell's DNA and the vital proteins during the adverse environmental conditions. Like what? Like excessive heating, drying, radiation, low nutrient content, etc. So these spore formations help the bacteria to remain viable for a long period of time even in adverse conditions. Okay. So this is what the endospore staining does. So as we know, this is the reason behind the formation of the spore coats or spores. Now this endospore which is developed, this is structurally and chemically a more complex structure, and it is very complex than that of the vegetative cells. Okay, now externally if you see, there are more layers. Here in this picture we can see there are so many layers inside, right? Now as the layers goes on increasing or goes on forming, this calcium ions increases, the calcium ion production inside the spores, it increases, and thereby increases the synthesis of the dipicolinic acid. Okay, now this dipicolinic acid, it imparts the trait that make the endospore chemically unique. Now this very dipicolinic acid is responsible for this, the resistant nature of the endospore. Okay, here in this image as we can see, there are so many layers. First there is the outer membrane here. Okay, this is the outer membrane, this is the exosporium, this is the outer membrane, here the cortex, then the inner membrane, then there is a germ cell wall, spore coat, core, then inside it there is a core. Okay, there are so many layers we can see, and inside all of the layers are protecting one portion, that is the core. Now this core contains the cell's DNA and the vital proteins that are required by the bacteria to become viable again after the adverse condition is over. Okay, so this is the reason behind the resistant of this endospore, behind the resistant nature of this endospore, and this makes the endospore staining usable. Okay, only endospore staining is used for this purpose, otherwise other staining are not developed, are not used for this for to stain the endospore, they are not applicable to stain this endospore. Okay, so this is for the endospore staining, this is the reason. Now the capsule staining, what it does, This depends on the charge of the cell wall. Capsule staining, it's basically depends on the charge of the cell wall. As the use of the acidic stain, this creates the repulsion between the negative charges of the stain. Now for the capsule staining, we use the negative staining or the negative dyes, and since the bacterial surface is already negative in nature, already negative charged, right? So bacterial surface is negative in charge. So that is why when we use this acidic stain, it creates a type of repulsion between them, which renders the dye to not penetrate inside the cell. Now here in this picture, you can see that the bacterial cell wall has all the negative charges, and when we give the dye who have negative charge, they will repel each other, and that is why the bacteria do not take up the capsules do not take up the dye and appear as clear halo region. Okay, so this is the complete mechanism of the endospore and capsule staining. Now the details of this mechanism is also covered in the endospore staining and negative staining video. The links are in the description. If you want, you can go and check it out. Now coming to the applications of this endospore staining and capsule staining. Okay, so first the endospore staining. This helps to identify endospore forming bacteria and assess their survival mechanism. Okay, this staining will also assess their survival mechanisms. As I said, in the adverse condition, they will become dormant using this endospores, and after, when the conditions are favorable, they will again regain their viability. Okay, so this is the study of the survival mechanism. Second, this staining method also determines spore production for disease, survival and virulence studies. Okay, so this will also allow to determine the spore forming bacteria, which may be again analyzed for the disease production, for the survival strategies and also for the virulence studies. Now, capsule staining, on the other hand, is used to observe the intact microbial structure without disturbing its cellular morphology. It allows the visualization of shape and size of the organism against a dark background. It observes the bacteria that are difficult to stain. Now, capsule staining uses the negative staining. So, these capsules are very difficult to stain using the other dyes. So, that is why the negative stain or negative dyes are used for this capsule staining. Now, this staining also helps in identifying encapsulated pathogens and understanding their role in infection, virulence, and immune evasion. Okay. Now, this capsule, most of the capsule forming bacteria are pathogenic in nature, and this staining method helps to identify these encapsulated pathogens. Okay. Now, let us have a quick review of what we have learned, the summary of this staining. So this is the whole summary. So the purpose of the endospore staining is to visualize bacterial endospores. These endospores are dormant and resistant structure. Then in the capsule staining, the purpose of the capsule staining is to visualize the bacterial capsules. This capsule is a protective outer layer which is usually made up of polysaccharide or a protein layer, and this capsule is present around the cell. So this is the purpose. Now the principle of the endospore staining is that these endospores are highly resistant structures with tough outer membrane composed of keratin-like proteins, which makes them impermeable to most stains. So this is the principle of endospore staining. For capsule staining, on the other hand, these capsules are gelatinous outer layers that are non-ionic, which do not allow them to readily bind with acidic or basic stains. Okay, since they are non-ionic, they will not allow to readily bind. Now, coming to the staining technique. So here, for endospore staining, Schaffer full tone staining is used. It's a type of differential staining, as I have said earlier. And for capsule staining, the staining method is negative staining. Okay, the stains that are used for endospore staining are malachite green and safranine. Malachite green is used as primary stain, whereas safranine is used as counter stain. For capsule staining, primary stain is India ink or nigrosin, and the counter stain is crystal violet. Okay, the mechanism is for endospore staining, heat is applied to force the malachite green into the tough keratin-like spore coat. Endospore retain the primary stain after cooling, whereas the counter stain it dyes the vegetative cells and appear pink. Right. For the capsule staining, the mechanism is that the capsule is not directly stained due to its non-ionic nature. The background and the bacterial cells are stained, which leaves the capsule as colorless halo region around the cells. Okay, so this is the mechanism. So here if we look for the modern, so for the endospore staining, heat is used as a modern. No other reagent is used, only heating is used. As I've said in this pictorial flowchart only, the heating of the smear after giving the malachite green is very important step in endospore staining. Okay, so here the heat is used as a modern to drive the stain into the endospore. And for the capsule staining, no heat is required, no fixative is required even. No heat or fixative is applied to avoid destroying the capsule. As the capsule, when the heat is applied, the capsule will get destroyed. So to avoid this capsule capsule hindrance, what we do, we generally avoid the heating process. So it is not required. So it will keep the structure of the capsule intact. Now, for the target structure, for endospore staining, the target structure is endospore, and for capsule staining, the target structure is obviously capsules. And what are the outcomes? For endospore staining, the outcome is that the endospores will appear green, whereas the vegetative cells will appear pink. And in capsule staining, we can see clear halos around the stained cells. Okay, so this is the quick recap of the endospore staining versus capsule staining. I hope you all have understood this video. If you have any queries or any suggestions, you can give your comments below. And if you have liked this video, do give a thumbs up and do like, share and subscribe to our channel for more such related microbiology updates. Also, for details of this video, if you want the details of this video, you can check our LinkedIn page. The link is in the description as well. And I'll see you in the next video.